Hello all, welcome to the video on the visual system. This is the last of the senses we're going to talk about, and the visual system covers the sense of sight. So, again, visual system, sense of sight. Now, all the stimuli for the visual system is light. So, light are a form of electromagnetic radiation that travels as a wave. In other words, we're talking about like a light beam. Light actually travels in a wave, similar to the sound waves. So, similar to sound waves, light waves can also vary in amplitude and wavelength. So amplitude will affect the perception of brightness. So again, you can think of that as the volume. So the volume of color or how bright the light is. And then wavelength will affect the perception of color. So whereas before wavelength and frequency was for pitch, so different pitches of sound, so high voice versus low voice, wavelength affects color. Now, humans, we also see varieties in purity of color. So this influences the perception of saturation, or how rich a color is. So not brightness, but richness. So this is just a picture showing all the different gamma radiation that there is. And so really you can think of this as the light waves. There's a very large range of what light can do, or what wavelengths it is. But only a very small portion, kind of in the middle here, is visible light. So we can only see this small amount of the light rays. And this is from wavelengths of about 350 to about 800. So we really can't see too much of it. Now, lower light waves would be the cooler colors, so like purple, blue, and then into green. And then the higher the wavelength, or the longer the wavelength, then the more red the color, the warmer the color. Okay? So, there are ultraviolet wavelengths that you've probably heard of. So ultraviolet is the wavelengths kind of just outside of our visible light spectrum on the cooler end, so like a purplish color, and that's why it's ultraviolet. And then the opposite of ultraviolet is infrared. So again, that's just outside the visible light spectrum, but on the red side. So that's why it's infrared. Now you can see that the light spectrum also covers radar, radio signals, TV signals, as well as on the lower side X-rays and gamma rays. All of this is made up by light rays, or as we call them, gamma rays. But again, we can only see a very small amount. Now, how do we see? Obviously, we see with the human eye. Now, this is a great diagram because it labels everything you're going to need to know. Okay? So I would really study this image and be able to label everything in it. So, starting from the back of the eye over here, we've got the optic nerve. So this takes the information that the eye sees and brings it to the occipital lobe in the brain. Also back here, we have the fovea, the retina, the blind spot. We'll talk about all those. And then in the front of the eye, we've got the lens. We've got the muscles that help control the lens. We've got the pupil, the cornea, and the iris. So that's a lot to talk about, so let's kind of break it down. So the eye, their job is to channel the light from the outside world to the retina. So the retina is really this broad back area of the brain. So the light will enter through the cornea. So the cornea is the clear covering over your eye. So when you touch your eye, you're touching the cornea. And what the cornea does is it helps focus the light to the lens. So all the light from the cornea then goes to the lens, which focuses it. And then it helps focus it so it goes back to the retina. Okay, so light passes through the cornea to the lens where it's focused onto the retina. Now the lens itself can move or change because that's how it focuses. And so we call these adjustments accommodations. So this is when you're focused on a close object versus a far away object. So when you're focusing on a close object or something closer to your eye, then the lens of your eye will get fatter or rounder. So pretty much the muscles kind of, they elongate so that the lens is nice and round. And when you're focusing on something farther away, so if the butterfly was way over here, then the lens will flatten out and give you a better image. And it does that by the muscles will contract or get shorter and kind of pull the lens on both areas. Okay? So both sides will go out and it causes the lens to get flat. And that way you can focus on something farther away. So the eye can also adjust the amount of light. So by changing the lens, that's how you change how you focus. But you can also change the amount of light. So if you're in a dark room or a light room, your eyes will change a bit. You might have noticed some, they say your pupils contract or expand based on the amount of light. So the pupil is really, there isn't really a thing that is the pupil. The pupil is actually just like a hole. Okay, so the pupil is the black center of the eye. So when you're looking in the mirror, then it's that black center of your eye, and there really isn't anything there. So the pupil is really the gap between your iris. And so the iris is the colored muscle around the pupil. So it's, that's, your blue eyes, your brown eyes, your green eyes, hazel eyes, whatever, that's the iris. And that is a muscle that is in charge of regulating the amount of light that gets into your eye. And it does this by changing the size of the pupil. Now, we're going to spend a bit more time on talking about kind of the inner eye. So we talked about the outer eye, or the stuff you can see a lot now. So let's talk about the stuff in the back. So big one of the main parts of the eye is your retina. So the retina is neural tissue lining the inside back surface of the eye. And it's super important because the retina absorbs light, processes the image, and then sends the image to the brain. So it's very important. Now, one thing I do want to point out is that everything you see, your eye flips it upside down. So here, the top of the wing here, it gets focused on by the lens and gets sent to kind of the bottom of the eye, whereas the bottom part of the image gets focused by the lens and sent to the top of the eye. So your image ends up upside down. Okay? Now, it's the brain's job to flip the image right side up. So everything the brain gets is actually upside down, but it flips everything. Okay? Now, within the retina are multiple different types of cells. So if we start over here on the right side, that is the back of the eye. Okay, so that's here. So it's actually kind of backwards. So back of the eye is here. And here we have two types of cells, either cones or rods. And then they connect to other types of cells that help process the information. Okay? Now, in this image, you're probably confused because it says light is here, right? It says the light is coming from the left side, but I just said the right side of the image is the back of the eye. All right? So light actually goes past all of this and is then processed by the cones and rods first, and then is processed by this. So the light goes past everything to be processed, and then works its way to be processed by these more specialized cells. So you don't need to know this too much. This is just a fun image. You'll talk about this more in a second year course if you take that. But I just wanted to show it. For this class, we're only going to focus on cones versus rods. Okay, so you need to know this image of the eye, and then what cones versus rods are. And again, we're going to talk about that here. So cones, I'm just going to show you this image again. If you see here, they're the red ones. They actually are conical in shape, or they look like little cones. Whereas rods don't. They just look like rods. So they're named after what they look like. So cones specialize in visual receptors that play a key role in daylight vision and color vision. In other words, cones, I like to think of C for cone and C for color. So cones process or are the receptors for color. We also tend to use cones more in the day because they don't require, or because they require more light, and we have more light during the day. So when it's light out, you're using your cones more. So because the cones are more important during the day, and they're specialized in color vision, which means more detail, then the cones tend to be concentrated near the middle of the retina. So if we say the retina, I'm going to draw again, guys. Let's use this fun color. So the retina starts here and kind of ends here, right? And it covers all of the back area. Then our cones are going to kind of be focused within this section. Okay? Or like a little like that. And you're going to have the most cones here in the middle, and it's going to slowly kind of taper out. Or you can think of that like normal distribution, like that. Now, if this doesn't make sense, that's fine. I have an image of that. But pretty much, we have a lot of cones in the middle of the retina, and not so much on the edges. Okay? That's really what I want to talk about. Now, if we erase all that, then now let's talk about the fovea. So the fovea is right here, okay? And there's kind of a little dent in the retina there, okay? And that's really just so that you can see where the fovea is. And what the fovea is that is a tiny spot in the very middle of the retina that contains only cones. Okay, so you only have cones and no rods. And that is the spot of your eye that will have the sharpest image. So really everything is trying to be focused by the fovea. So that's like the focal point. Okay, so fovea is like the focal point. And because cones contain more or help with detailed information, then there's only cones in the fovea. And that's why next to the fovea, again, is where you'll see the most amount of cones. And then as you get further away from the fovea, you see less and less cones. Now, the opposite of a cone is a rod. So rods are a lot more common outside of the fovea or on the edges of the retina. But there are a couple near the fovea, but remember there's none in the fovea. And it's specialized in night vision and peripheral vision. So seeing things that aren't what you're focused on. And then also night vision because it's less detailed and it's more about light. So rods don't need as much light to work as well, and that's why they're good for night vision because there's less light. Now, I told you I'd show you a fun image. So this is showing us what the amount of cones versus rods look like when you're looking at the fovea. So here's the fovea, and we're calling that 0. So the angle of degrees is spots away from the fovea. So the further away from the fovea, the bigger the angle. So at the fovea itself, you can see there's a huge peak of a whole bunch of cones. So red is cones. So right here, there's a huge peak of cones. And then going to about here and here, so that's like right here on the graph, you also have a pretty decent amount of cones. But then anywhere else, there's almost no cones anywhere else here, right? And that's what the graph is saying. Whereas with rods, there's rods all over here, up to about here, and there's rods here. But then when we get about 10% or 10 degrees away, then suddenly it drops off and we don't have any more rods. So when you're closest to the fovea, you'll have lots of cones and not a lot of rods. When you're away from the fovea, you'll have lots of rods but not a lot of cones. I'm going to stop there for this video, and when we come back, we're going to talk about different adaptions and color.